Let us deal with the tertiary structure of a protein. Tertiary structure is nothing but the spatial arrangement of amino acid. Now remember, if the spatial arrangement will be such that it will have minimum energy and maximum stability. It will have minimum energy and maximum stability. And this tertiary structure is also for having a particular function. So if you denature a protein and try to assemble it n number of times, there is a possibility that there can be n number of possible arrangements. And all these, it may not fold in the same manner each time. And this folding, one with the minimum amount of energy which gives the maximum amount of stability, is the one that will be native and that will survive. This is through a process of evolution. Nature has done this process to ensure that the most stable form of the protein will be the one with the minimum amount of energy. This is something that is proved with the Unfinson's experiment also. Now let us see which are the forces which will ensure that this minimum energy is present and there is maximum stability and the right conformation of the protein is established. There are 6 different types of forces which will ensure that the protein is stable. First of all, there will be covalent bond. That will be either disulfide bonds which is present in the protein. Now these disulfide bonds may not be present in all the proteins. Only those proteins that has got a presence of cysteine residues. So in other proteins the disulfide bonds may not be present. The most important non-covalent forces which will stabilize a protein, first of all we can say that in the bond that is formed, ion-ion bonds, ion-ion interaction, ionic interaction, that is between the R groups of the amino acid, that has got, let us say, a positive charge with the other amino acid which has got a negative charge. For example, you can have an arginine that has got a positive charge that is interacting with the aspartic acid which has got a negative charge. You can also have lysine which has got a positive charge interacting with the glutamic acid. This is one possible interaction that will stabilize the tertiary structure of a protein. In the second kind of interaction is, as you know, there are plenty of hydrogen bonds that are present. Hydrogen bonds which are present between the alpha helical structure and the sheet structure will stabilize the tertiary structure of a protein. In the third, so hydrogen bonds. In the third kind of interaction is, in many cases there will be metal ions, divalent cations like manganese, magnesium, cobalt, zinc, etc. These metal ions will interact with negatively charged amino acids like glutamic acid, aspartic acid, etc. and that will try to stabilize the structure of a protein. The fourth kind of non-covalent interactions are the ion dipole interactions. The ion dipole interactions can be, for example, if you look at glutamic acid, it will have a negative charge. So this negative charge can interact with a slight positive charge of certain amino acids like threonine, etc. That is one possibility. Or in the carbonyl groups that will have a slight negative charge, this can interact with the delta positive of lysine. So these are examples of ion dipole interactions. In the fifth kind of interactions are the hydrophobic interactions. You know that the hydrophobic interactions is the tendency of this the amino acids to remove water from its surrounding. Especially the hydrophobic amino acids will come together like isoleucine, valine, all these phenylalanine, all these amino acids will come together, they will club together, remove water, and in such a way so that they form a tight, compact core structure. This is another interaction which is present in the tertiary structure or which will contribute towards the formation of the tertiary structure. The last kind of non-covalent interaction is the non-peptide dipole interactions. So that is a non-peptide dipole interaction that you will see in the imidazole group of histidine and in the hydroxyl group of serine. This is another type of interactions. So these are the 6 different types of non-covalent bond, and we have seen one covalent bond, that is a disulfide linkage. All these will make the tertiary structure stable and give rise to a stable protein with a minimum amount of energy. Always remember that the tertiary structure is formed with a particular function, which is the functional enzyme forming, and that combinations will be promoted by these non-covalent as well as the covalent interactions.